hear me? I'm not hearing anything. Are you guys hearing me?
Yes.
Oh, thank
Yes,
you.
I
Yes.
can hear you.
All right, let's begin then. You should be able to see my screen. Let me talk just a little bit about the syllabus, the material for the lab. There is no lab textbook. Everything for the lab will be online. Reading material too. The lab is an integral part of the course. Microscope use in virtual lab. You're not going to be using microscopes because we're not going to go into campus with the microscopes, but you will be expected when we cover microscopes to learn the parts of the microscope and how they work. We will have some virtual labs. Many of the virtual labs use Flash, which is no longer supported. Because of that, we pulled those out of the lab and we tried to find alternatives, but it was difficult finding alternatives. So we only have, I think, 2 virtual labs where you will be doing the lab work using your computer. Like with the microscope, you will actually be adjusting the microscope or working on a virtual microscope on your computer where you can adjust the microscope, look at a slide, adjust its viewing. The point of the virtual lab is it does allow you to do some of the activities that you would normally do in the lab on your computer. We do have a few virtual labs. Everything that we covered applies. I think I'll skip that. We can read about it. We do have lab safety rules. Normally, these are rules to keep you safe while you're in the lab. You're not ever going to be in the lab, but we do require you to know these safety rules because safety is one of the main skills you will need to learn while you're taking microbiology. It's also a skill that will help you very well when you go into your medical world. Like nurses need to know how to be safe when they're handling specimens from a student, from a patient. Oops, what happened there? I don't know why that came up. Yeah, there it was. My computer is acting a little slow because it's still converting the lecture. It's only 72% done. About the lab, if we were to come into the lab, all personal items, including your book bags, your books, your purses, your cell phones, your coats, must be stored in a cabinet to store them. We call it a cubby hole. And you put it in there. And the door and the cubby hole are not considered part of the lab. It's considered outside the lab. And then when you walk away from the door and the cubby hole, you will go and wash your hands put on your, let's see, you put on the lab coats first, then put on the gloves, and then put on maybe goggles. Take off the goggles, take off your gloves, take off your lab coat. Yeah, that's the right procedure. And the point is you walk over to the sink, wash your hands. That is considered part of the lab, and everything that's in the lab stays in the lab. So if you were to bring a pencil into the lab, that would be fine as long as it stays in the lab. Once you bring it into the lab proper, and that's beyond the door and the cubby hole, it then stays in the lab permanently until it is sterilized. Once, like example, you want to take the pencil home, you have to turn it into your instructor, who will then get it sterilized, probably by autoclaving, and then we'll return it to you, and then you can take it out of the lab. The point is anything that comes into the lab stays in the lab. Now for usually pencils, pens, and paper, we give you those in the lab, and they'll stay in the lab. They never leave. If you wanted to take a copy of your notes home, you would have to take a picture and then email the picture to yourself, and then you could take that home, either the picture in the email, or you could go to someplace and print it out and take it home. But the point is, stuff that's in the lab stays in the lab. And the purpose is to keep you and others safe. We don't want the microorganisms which are in the lab to leave the lab. Any questions about any of that? So generally speaking, stuff is not brought into the lab, although you could bring in, like I said, a paper and pencil and things like that. And as long as they stay in the lab, that's fine. They cannot come into the lab and then leave the lab. If you do have it in like your backpack, you just leave it in your backpack, put that in the cubby hole, and we consider the cubby hole outside the lab. And then that can go out. Cell phone use is prohibited. If you were to use your phone in the lab, it would be confiscated, and then it would be sterilized, which would require to be dunked in a disinfectant like bleach or stuck in the autoclave. Either way would destroy your phone, and then you could take it home. All cultures are potentially pathogenic that you work in the lab. They obviously never leave the lab. You should keep your hands, pencils, and other materials out of your mouth, your ears, your nose, etc. No eating or drinking or smoking or gum chewing or any oral habit is allowed in the lab. You must wear gloves. They are put on shortly after you enter the lab. So after you come into the lab, you put your books in your bag and your coat and whatnot in the cubby hole, and that's considered out of the lab. And when you enter the lab, you go to the sink, you wash your hands, you do it the correct way, washing everywhere for 20 seconds, and then you dry your hands. You then go and put on your lab coat, which generally is kept in the lab. That never leaves the lab, although if you bought a lab coat, you may bring it into the lab, and then it stays in the lab. If you want to take it out at the end of the term, it would be washed, actually autoclaved and then washed, and then you could pick it up and take it out of the lab. Yep, looks like the lecture finally finished. Let me finish this. And now my computer will work a little faster. So you come into the lab, you wash your hands, you put on gloves, you put on your lab coat, you put on gloves, and then you come to your workbench, the place where you work. If you're going to be working with a splash hazard, you put on goggles. Most of the time, we do not work with a splash hazard, but if we do, you'd have to wear goggles. What you do before you begin the lab is you clean your lab bench with disinfectant. It's to remove all microbes from the area you work in, not only to clean it from perhaps the previous person who may have spilled bacteria and then never wiped it up, but also to decrease the contamination around you. And then you do your work, and then when you're all done, the last thing you do in the lab is you wash down the workbench area with disinfectant, wipe it down with a paper towel. I'll put that in the garbage. And then you go and you take off your gloves, take off your lab coat, go wash your hands, and then you go to the cubby hole. So now you're out of the lab. Grab your things from the cubby hole, and then you leave the door and leave for the day. So that's what you would do if you were coming into the lab. Any question about that? All right.
Is there any, there won't be any reason for us to go into lab on campus?
This term, we will not be going into the lab, but you do need to know the procedures for using a microbiology lab, and you will be quizzed on it, which is why I went over that procedure.
Okay.
Okay. While you're in the lab, you must wear closed-toed shoes, meaning a shoe that is totally covered. The main reason is to prevent a spill from getting on your shoes. Like women have fancy open-toed shoes, and if you were to spill microorganisms, they would get on your toes, and it would be hard to get it off. And so we do require you to have closed-toed shoes in the lab. And then you have a lab coat, which would help also. Lab coats must be worn and completely buttoned. And then the gloves help. Accidents have to be reported to your instructor. Minor accidents should be cleaned as the following. You guys can see this, right?
I can
You
see
guys
it. Yep.
Yeah, we can see it.
Great. Always clean broken glass first. Broken glass can cut your skin, and glass is a problem. Take the brush and the broom and the dustpan. Never pick up glass with your fingers, and dispose of the broken glass in the proper receptacle, meaning we have a receptacle for broken glass only, and put it in there. If you spilled strong acids and bases, they should be diluted and wiped up quickly. Spilled bacterial culture should be cleaned as follows. Cover the spilled material with lots of paper towels. Drench the paper towels with disinfectant. Let it stand for 15 minutes before wiping it up. Wipe it up with paper towels. Be careful if there's broken glass present. And then take the paper towels and the cleaned up spill and put it in the biohazard trash can. For the broken glass, it goes in the broken glass receptacle. And it is difficult pulling out the broken glass from the paper towel and the disinfectant, but the bacteria spill. But put that in the biohazard trash bag, which is also called the autoclave bag. All right, keep hair away from Bunsen burners and back to incinerators. I'll show you what a back to incinerator is in just a minute. Mouth pipetting is not allowed. Lab cultures may never leave the lab. If you have a cut on your hand, wash your hands, apply a bandage, then put the glove on. All microbial cultures must be completely labeled with your name, the date, the nature of the specimen, and the medium used. And it's optional to put the temperature grown on. Specimens, bacterial specimens must be discarded properly in the following ways. An agar plate will have a petri dish made out of plastic to put it in the biohazard container. Glass slides are put in the glass container, cotton swabs in the swab container, and those are at the back of the lab. Pipette should be placed in the appropriate pipette container when you're done using them. Glass test tubes should have the tape removed from them and placed upright in the test tube racks in the discard bin near the rear of the lab. We don't really use glass containers, but if you would, those would be put right next to the test tubes. The test tube generally is the only glass container we use. All other bacterial specimens, like something you spill, you wipe it up with paper towels, the agar plates, put those in the biohazard container. Supplies needed for the lab should be at the front of the lab. Remember that where your books and backpack are kept is considered outside the lab. And so you should stay away from that area while you're working with stuff in the lab. And you shouldn't go to that area except when you first come into the lab and when you're about to leave the lab. When you leave the lab, as I stated, wash down your lab bench, remove your lab coat, remove your gloves, wash your hands, and then leave the lab. So you're required to follow and know these rules, even in this virtual lab. You will be quizzed on some of this material. All right, any questions about the lab? That's it for the syllabus for the lab. Let me state that we're going to do one lab today. Lab 00 is going to cover lab safety training. You need to read the infectious disease research project file, which you can find in Canvas. You need to have that read by next week because we're going to talk about the infectious disease research project next week. You can find that. There it is, week 2 of the lab infectious disease research project. Any question about any of that? All right, let's talk about lab 00 then. Any questions? Go here. So when you're working on the lab, there is both a lab module and a lab worksheet. That's the worksheet. That's the lab module. You can read the lab module beforehand. In fact, you can do the lab beforehand, but we'll talk about the lab module. And I grabbed the right one. I think I grabbed the right. I don't seem to have it. All right. So you're not going to be working in the lab, but you do need to know this material, as many of these rules and procedures will be relevant during your career, and you will be quizzed on some of this material. So the primary learning objectives for this lab is be able to distinguish between safe and unsafe laboratory practices, understand where relevant safety equipment is located in the lab. And for the online lab, really all you need to know is what is some laboratory safety equipment which is located in the lab. Okay, you don't need to know where it is because we're not going to go into the lab. Understand how to safely dispose of contaminated materials. So laboratory safety is expected that microbiologists observe a number of rules to work with pathogenic microorganisms safely. You're not going to be actually performing any of these activities in the lab, but it's important to understand the guidelines to create a safe laboratory environment. Safety precautions include wearing proper attire, which includes once you come into the lab, you put on a clean lab coat, which covers your lap when sitting. You wear disposable gloves and you have closed-toed shoes on. If you're working with a splash hazard, you wear goggles. Okay? Now, what of these 4 things do you put on before you come into the lab?
Closed-toed shoes?
Say again? I didn't hear you.
Closed-toed shoes?
Yeah, so in the question it asks, what do you put on before you come to the lab? And what do you put on after you come to the lab? And you put on closed-toed shoes before you come to the lab. What do you put on after you come to the lab, meaning after you enter the lab?
Lab coat,
Lab coat's one, what else?
gloves,
Gloves.
Gloves. And if you're wearing goggles, you put those on after you enter the lab. There are 2 questions on the worksheet today. One asking, what do you put on before you enter the lab? And what do you put on after you enter the lab? And there's only one word different, before and after. Okay, but I've already told you some of the answers. What's something else you put on which helps with safety before you enter the lab? And that is not covered in this lab manual, which is why I'm asking you now.
Is that your hair tied back?
Well, you could put on a hair tie to tie your long hair back. That is covered here with something that you put on for safety, kind of. You put it on for other reasons, but when you're going into the microbiology lab, it does help keep you safe.
Pants.
Yeah, I was
Say
gonna say
again?
pants.
Pants?
Pants. Yes.
Yeah, that's true. Clothes is all you needed to say, but pants would be correct. Yes. Okay. All right. So that's another thing. I think when you put on the question, let me go look at it beforehand. I think there's only 2 things you need to answer on that. Nope, there's actually 4 things. So you have to come up with 2 more things. All right. Long hair should be tied back. Absolutely no food or drink in the lab or any electronics. Anything like that would be confiscated from you, and you wouldn't get it back until it's sterilized, and that would destroy the electronics. To tell you the truth, most food items would be destroyed also. So no personal items are allowed in the main part of the lab. You may bring things in the sense where you put it in the cubbyhole, and we consider that outside the lab. Anything brought into the lab, you cannot get back until it's been sterilized, like soaking in bleach for 15 minutes or stuck in the autoclave. When you come into the lab, no food, no drinks, no cell phone, no headphones. And you should look like this, where you have a lab coat on, gloves. Our lab benches are not quite looking like that. And then closed-toed shoes. I think that's supposed to say the lab coat's buttoned. I'm not sure if she has goggles or those are glasses, but if you're working with a splash hazard, you would wear goggles. Keep your work area clean. As I stated, before you begin work, you should wipe down your lab area. And this is actually a picture of where we would work in the lab if we were going to the lab. Wipe down where you work, that'd be the work area here with disinfectant, and wipe it down with a paper towel. And you just throw that paper towel in the trash can. And then when you're all done, you do the same thing, wipe it down with disinfectant, and then throw that paper towel in the trash can. It's important to know where you discard waste. You will be tested on this. Regular waste, that's just trash, no sharp objects, no contaminated objects, no broken glass, go in the trash can. Okay? If it's been contaminated with bacteria and it's no glass, you put it in the biohazard container, shown here, and that will have a bag in it, which is the autoclave bag, and we'll take this thing all out, put it in the autoclave, and autoclave it. That would kill the bacteria in it before we then throw it away. Okay? Anything that's glass never goes in the biohazard contaminant area. Glass, like test tubes, go in the test tube rack. Remove the tape from it. Soiled swabs go in a swab container where there will be bleach in it. Slides go in the slide container where there will be disinfectant or bleach in it. The cover slips we do not keep, but they go in a separate container. Okay? Meaning we will disinfect those and then throw them away. The slides are actually disinfected and then they're washed and then they'll be reused. The swabs are put in disinfectant and then they'll be thrown away. Consider all organism you're working with to be potentially pathogenic. If you're unsure of how to dispose of something, ask your instructor. Report all spills and accidents immediately to your instructor. Cover it with paper towels and then saturate it with disinfectant. Allow the disinfectant to soak for 15 minutes and then wipe it up with paper towels and put it in the biohazard trash container. The exception is if there's a broken glass or glass in here, the test tubes would be put in the test tube rack and the broken glass in the broken glass container. All reagent bottles as well as organisms should be kept covered when not in use. Don't pour chemicals down the drain without getting permission, and generally do not put chemicals in the trash. No mouth pipetting. We have a bulb to work with pipettes. Don't stick things in your mouth while you're in the lab. We use a Bacto incinerator to sterilize your tools you work with. This is a Bacto incinerator. And as you can see, the stone on the inside gets red hot. When you're not using the Bactol incinerator, turn it off. If you're going to use the Bactol incinerator, you generally want to turn it on about 5 minutes before you're going to use it, because it takes about 5 minutes to warm up. So generally, when students come into the lab, they'll turn on the Bactol incinerator, wipe down their lab bench, and wait for the instructor to do their little talk, like I'm doing now. And then when they're ready to work, the Bactol incinerator will be red hot. We don't actually use a Bunsen burner in the lab, and we would not unless for some reason the Bactol incinerators would not work, like we didn't have electricity. Generally, though, if we didn't have electricity, I wouldn't think we'd be even in the lab working. But we do have the gas Bunsen burners for a backup. I've never used them in all my years at Clark College. So you should know some of the lab safety equipment, such as an eye wash station, shown here. Like if you get something in your eyes, you come to the eye wash station to wash it out. You get something on your body and you want to wash it out, you come into the deluge shower. And I should warn you that if you were to use that, that just gets this water on the floor here. It's true we have a drain underneath to collect the water, but the whole area will be wet. Fortunately, I've never had a student needing the deluge shower. I think I've only once in all my years of teaching had a student use the eye wash station. We have a spill control station. So if you spill something, you can use this to wipe it up. A fire alarm. We have a fire extinguisher in the lab and a fire blanket, which isn't shown, although it's stated here. There's a first aid kit. So those are some of the safety equipment that are in almost all labs at Clark, certainly in all microbiology labs. We have a few other things like a chemical spill, like if you spill acids, there's something to neutralize the acid. And then if you spill a base, there's something to neutralize the base. All right. So complete the laboratory exercises on the worksheet. Do not complete it in the lab manual and then submit the lab manual. If you submit the lab manual, it's much bigger than the worksheet, and so you may get a penalty. I'll probably first give you a warning, do not submit the lab manual because it's bigger. It will fill up my Canvas web page. Submit the worksheet. The worksheet also may have had the questions updated, so it rarely may have a different question than the lab manual. So learn about safety and what safety and accuracy are. We already talked about that. I'm not going to talk about safety infractions because you're not actually going to come into the lab. You don't really need to monitor yourself because you're not going to ever handle bacteria. And we do have a document for safety information for patients who, not students who were to get sick when we're microbiology students, when you're handling microbiology students. It's not totally applicable to you guys because you're not going to handle actual bacteria. It probably tells you in the past that somebody once got sick who was a microbiology student, and the material was a bacteria they took home with them from the lab. And that's why we follow these safety rules. All right, I've already talked about that, so I'm not going to talk about it again. Keep hair and paper away from the Bacto incinerators. All right, I think that's it. Any questions about the lab? If not,
I just have a question about Rule 5.
let's go to the, oh, I thought I shut that down. Let's go to the worksheet. So in the worksheet, you can actually skip the first page because the questions don't come out unless you print it. Don't ask me why it doesn't show, but I've got the questions here. When you're answering the questions, wherever there's an asterisk, means you need to have that many answers. So for Rule 1, you need 4 answers to answer Rule 1. In the lab, what do you always wear for safety before you enter the lab? For Rule 2, after you enter the lab, what do you put on to keep yourself safe in the lab? And you only need 2 things there. For Rule 3, you have to look up what is a safety data sheet online and then state what each chemical in the lab must have a safety data sheet that describes what. And there's going to be 1, 2, 3, 4, 5 different items you list. Generally speaking, you have to put in more than 1 word. Like if you say chemical, you have to state what chemical. And if you say safety, you have to say a little bit more, like how to keep yourself safe from this chemical. Okay, so generally 1 word answers don't work. Know the location of the safety equipment, and you have to label 1, 2, 3, 4, 5 different safety equipment we have in the lab. And then somebody had a question about lab 5, Rule 5.
Yes, so what you can take into the lab, you want 2 things, and then can you eat or drink in the lab? Is that like a question that you just put yes or no for the last one?
For the last one, you just put yes or no. And what can you take into the lab? You got to think about it. Like, for example, I've got reading glasses on. Can I take glasses into the lab?
Yes,
Yes, I can. If you were wearing a hearing aid, could you take a hearing aid into the lab? Come on, people.
yes,
Yes. If you're wearing a watch, can you take a watch into the lab? Yes. Okay. So there are some things we do allow in the lab, but generally, any personal items we do not allow into the lab. But there are a few things. Okay. And I just went through and told you some of the answers for what you can take into the lab. I didn't tell you this one, but that should be pretty obvious. And then list one item of laboratory attire you must wear in a microbiology lab. This one can be a repeat of Rule 1 or Rule 2. List one item that is allowed in a microbiology lab. And this could be a repeat of Rule 5. List one item that's not allowed in a microbiology lab. And you could put something like my Big Mac sandwich. Just trying to be a little funny. My ice cream cone, okay? There's a lot of things that are not allowed in the microbiology lab.
For most of these worksheets, do you just type on to the worksheet itself and then save it and turn it in?
That would be the best way.
Okay.
I have had students print it out, write in their answer, take a picture of it, and then submit it. It's much more work, but you could do it that way. It'd be easiest if you just typed in the answer in the worksheet and then submit the worksheet. All right, for lab 00, it must be submitted by 8 a.m. Friday of week 1. in the folder in Canvas for worksheet lab 00. Or you may be dropped from the class. I'm going to use this worksheet for attendance for who is attending my class. This will be the only time I do this. Any question?
I do have a question about number on rule 3. I found 16 things for that, and there's only 5. So is there a specific place that we're supposed to look, or?
I'm sorry, it's only asking for 5 things. You went and looked up safety data sheet online?
Yeah,
There's got to be much more than 5 in the safety data sheet.
yeah, there's at least 16 from what I'm looking
Okay, you
at.
only need to put 5.
Okay,
Okay. That's what I misheard. There's 16, you said, okay. All right. All right, in the lab, I will be here to answer any questions. I will be here until 8 o'clock. And then I will leave at 8 o'clock. However, if the last student logs off before 8 o'clock, I will log off at that point. So if you have a question, make sure you ask it before the last student logs off or before 8 p.m. Any questions?
So then is lab kind of independent and then we just kind of stay on Zoom and ask you questions if needed?
Yes,
Okay, and will it be like this for most labs?
you will be like this for all the labs.
Okay, okay. Thank you.
Well, wait a minute. For all the labs where there is a lab, there are some weeks where we will not have a lab module, in which case it'll be different. I'll be just taking questions and answers. But for all the labs where there is a lab module, they'll be like this.
Okay, thank you.
Okay. All right. So begin working on the lab. I'm going to stop recording. And to do that, I have to stop